Hello students, I am teacher Svetlana and in our today's lecture we will learn about the cell cycle and mitosis in the chapter cell division. So let's begin. Students, the life of all multicellular organisms starts from a single cell, that is the zygote. Growth of every living organism depends upon cell division. As stated in the cell theory, every cell arises from the pre-existing cell. Cell cycle, students, is the sequential events occurring in the life of the cell, and that is called as the cell cycle. Cell division, DNA replication, and cell growth have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genome. The sequence of events by which a cell duplicates its genome, that is its genetic material, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. Phases of the cell cycle. The duration of the cell cycle can vary from organisms to organisms. For example, typical eukaryotic cell divides once in approximate every 24 hours, whereas yeast cell divides only in 90 minutes. The cell cycle is divided into two basic phases. Have a look at this image here. This is your cell cycle, and the two distinct phases are the interphase and we have the M phase. This is your pink line is your interphase, and here you have this blue line which is your M phase, which is also called as the mitosis phase. The interphase is divided into three further phases. G1 phase, that is gap 1. This is the G1 phase here. Then we have the S phase or the synthesis phase. This here is your S phase. And then we have the G2 phase, which is the gap 2 phase. This phase is marked here. The M phase or the mitosis phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. The M phase starts with the nuclear division corresponding to the separation of the daughter chromosomes known as karyokinesis, and it usually ends with the division of the cytoplasm known as cytokinesis. You can see this mitosis is further divided into a number of stages. Each of this stage we will see in detail ahead. So now students, we've just learned that the cell cycle has two distinct phases, the interphase and the M phase or the mitotic phase. Now, first we will have a look at what happens in interphase. The G1 phase corresponds to the interval between mitosis and initiation of DNA replication. So this here is your G1 phase. So before it's a time between the mitosis, the previous mitosis, and the next synthesis of your DNA or the replication of your DNA. So G1 phase, the cell is metabolically active and continuously grows, but it does not replicate its DNA. During the S or the synthesis phase, the amount of the DNA per cell doubles, but there is no increase in the chromosome number. So here you have your DNA replication taking place, the amount of the DNA doubles. So G1 you can consider as the preparatory phase where all the necessary preparations for the replication of DNA occur. In animal cells during the S phase, DNA replication begins in the nucleus and the centrioles duplicate in the cytoplasm. Then we have during the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. So here in your G2 phase, you have protein synthesis occurring. Now these are the ones which will play an important role in the process of mitosis. So cell continues to grow. The cells that do not divide further exit G1 phase to enter an inactive stage called as the quiescent stage G0 of the cell cycle. Have a look here. If a cell does not want to enter the G1 phase or begin its preparation for its next multiplication cycle, it leaves the cell cycle and enters a G0 phase. Here it is inactive, it does not divide. In animals, Mitotic cell division is only seen in the diploid somatic cells, and plants can show mitotic division in both haploid and diploid cells. Students, let us quickly know what are the types of cell divisions. There are three kinds of cell division which are found in animal cells. They are amitosis or direct division, mitosis or the indirect division, that is meiosis, which is also called as reductional division. So the M phase, what happens in your meiosis as well as your mitosis, we will see further. But here you have this particular division which is called as amitosis. It is the simplest mode of cell division. During amitosis, nucleus elongates and a constriction appears somewhere along its length. This constriction depends and deepens basically here. You can see this is your cell. Then here you have a constriction that occurs, and this constriction further deepens. It starts deepening and it divides the nucleus into two daughter nuclei. This is followed by the division of the cytoplasm. So here your nucleus is dividing. Then after your nucleus divides, the cytoplasm also divides, which results in the formation of two daughter cells. This division occurs in unicellular organisms, abnormal cells, old cells, and fetal membrane cells. So this is called as amitosis. And mitosis and meiosis we will see in detail. Now students, let us have a look at the M phase or the mitosis. Since the number of chromosomes in the parent and the progeny cell is the same, it is also called as equational division. There are four stages: prophase, metaphase, anaphase, telophase. So let us see what happens in the prophase. Prophase is the first stage of mitosis, which follows the S and the G2 phases of the interphase. The centriole moves towards the opposite poles of the cell. Have a look at this figure here. You can see this is the early prophase where you have this centriole that is pure, and you have in the later prophase you will see that there is condensation of the chromosomes that start, the nuclear membrane starts to disappear, and you have your centrioles which move towards the opposite pole. So these centrioles move towards two opposite poles. Here they were together, here they move to two opposite poles. Chromosomal material condenses to form compact mitotic chromosomes. So here there is condensation. Chromosomes are seen to be composed of two chromatids attached together at the centromere. So you can see this is here the centromere, and the chromosomes are attached in the form of two chromatids. These two segments are called as chromatids, and the one which the center point which holds these two chromatids together is called as the centromere. Initiation of the assembly of the mitotic spindle, the microtubules, the proteinous components of the cell cytoplasm all help in this process. Then we have metaphase. Metaphase chromosome is made up of two sister chromatids which are held together by the centromere. Small dish-shaped structures at the surface of the centromeres are called as kinetochores, where the spindle fibers attach. Chromosomes are moved to these spindle equators and they get aligned along the metaphase plate through the spindle fibers to both the poles. Have a look at this image here. You can see these are your two sister chromatids. Now there is a central region which is called as the kinetochore, and here you have these fibers that are getting attached to it. These fibers are called as the spindle fibers. Their work is to hold the chromosome intact. This is your metaphasic plate. So why it holds it intact? So that at the time of division the chromosomes split equally. Chromosomes are moved to the spindle equator. So this is your spindle your equator, and it gets lined at this equatorial point. The plane of the alignment of the chromosome at metaphase is referred to as the metaphasic plate. So this line where they get aligned is called as the metaphasic plate. Students, it is also said that chromosomes are best seen at the time of metaphase. Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids begin their migration towards the opposite pole. So here you can see that they have got arranged in the metaphase and now they are getting splitted. They are getting pulled to the two opposite poles. The centromeres of each chromosome is towards the pole while the arms of the chromosomes trail behind. So the centromere region is towards the pole and arms are tailing behind. The next stage is telophase. Here what happens is the chromosome clusters are at opposite spindle pole and they decondense and they lose their individuality. So here you can see that they decondense, they loosen up and form these single-stranded structures. And now again, what happens is you have the newly formed daughter nuclei. Formation takes place. That is, you have the nucleolus, Golgi complex, and the endoplasmic reticulum. They reformed. So in the prophase, we had seen that this nuclear nucleolar membrane and the nucleolus disappeared. Now it is reappearing. Then we have cytokinesis. Cells divide itself into two daughter cells. So after the nuclear division has taken place, now you will have the cytoplasmic division occurring. In animal cells, a furrow develops in the plasma membrane, which gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Here you can see the cell forms a furrow here. That is, it starts contracting and the contraction deepens and it gets separated out into two daughter cells. In plant cells, wall formation starts in the center of the cell. The formation of the new cell wall begins with the formation of simple precursor called as the cell plate that represents the middle lamina between the walls of the two adjacent cells. So here you can see a cell plate comes in the plant cell. At the time of cytoplasmic division, organelles like mitochondria and plasmids get distributed between the two daughter cells. So it gets distributed between the two daughter cells, and here you have the cell wall which gets formed in the plant cell. Let us have a look at the significance of mitosis. As mitosis is equational division, the chromosome number is maintained constant. It ensures equal distribution of the nuclear and the cytoplasmic content between the daughter cells, both quantitatively and qualitatively. The heredity material DNA is also equally distributed. It helps in the growth and development of organisms. Old and worn out cells are replaced through mitosis. It helps in the asexual reproduction of organisms and vegetative propagations in plants. The process of mitosis also maintains the nucleocytoplasmic ratio. Although mitosis is very reliable process for preserving the genetic makeup of cells or organisms, it cannot introduce variation or new combinations of the existing genes. So this process is possible, that is introduction of variation and having new combinations created only when the cell undergoes the next type of division, that is meiosis, which we will see in our next lecture. Thank you students for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe and keep learning.